Thank you, Jason, for the introduction. Uh, I will start my presentation. I will share the screen. Is it visible?
Yes, it looks great.
Yeah. So I'm Sharandeep Singh Chahal, and I'm a PhD student at University of Arkansas. And I did this work during my master's. And the topic is identifying the distinct features of an imperiled habitat that supports the endemic Texas Screwstem, also known as Bartonia paniculata subspecies texana in Southeast Texas. So what are we going to learn throughout this presentation? We'll learn about the rare and unique plant species Bartonia paniculata subspecies texana, commonly known as Texas Screwstem. This plant is endemic to Texas, meaning it's found nowhere else in the world without this place. So we'll explore why this plant is considered rare, the various threats it faces in its natural environment. I'll guide you through its distinct habitat characteristics, highlighting key findings from detailed and well-structured field study. And finally, we'll conclude, we'll examine the results of our research and discuss the implications for conservation and future studies. So about Bartonia, Texas Screwstem, one is PAB, which means uh, a positive site, but the plant is absent. So this is the edge location, which is 20 meters away from the core location of the species where we see it. And then we have a negative site, which is a circular plot. Uh, it is an independent plot, but a geographically paired site where the target taxon has not been recorded in past 15 to 50 years. So basically, we have two plots where we see plant and two plots where we do not see the plant. So before going to the sampling plots, uh, let's review this. We have a pair of a positive and negative sites in 3 counties. And then in each county, we have two positive we have a positive site and a negative site with 2 plots where we see the species and 2 plots where we do not see the species. And then I'll explain what the circular plot is. So it is a 15-meter diameter circular plot with 9 1 by 1 meter quadrats in which we sample our data. So one of the quadrants were placed in the center and rest were randomly distributed. So the central location was was the place where we see the highest density of plants, either during the sampling year or during the past year. So within each 1 meter plot, we recorded vegetation percentage, vegetation cover percentage, in which we included understory vegetation, including forbs, graminoids, saplings, ferns, and bryophytes. And then in uh, we did the canopy openness percentage. So we used a fisheye camera app and recorded this data during the noon at all sites. And later the data was processed with the ImageJ software. And next we see and record the ground cover percentage, which included live vegetation, dead rooted vegetation, organic debris, water, and bare soil. And then within each 1 meter square plot, we measured soil moisture using a soil moisture sensor and data logger. So at the whole circular plot level, we recorded overstory vegetation in which we included abundance of trees, woody shrubs, and vines. And then we recorded soil physiochemical characteristics, so in which we use one core of 10 centimeter was collected from each quadrant within each circular plot and mixed to obtain an aggregate sample for each circular plot. And later, the samples were analyzed at a soil laboratory at Lubbock. Let's go to results. So we have a total of 99 1 meters sampling quadrants in all sites. We pooled all the data and performed hierarchical clustering using Bray-Curtis dissimilarity matrix. And this gives us two groups of data showing presence and absence of target species. So the green color denotes the presence and red color shows the absence of the species in the plots. So this is cool that we see, our data is automatically grouped into two major groups where we see our species was present and where species was not present. So we used understory species composition data to carry out this analysis. And then we again did uh, did the same analysis with the larger 11 circular plots. So here we see, again, we see two groups, which denotes presence and absence, but we see some outliers. So the outlier lying in the present group here has a reason. So this was the most recent site that lost Bartonia in 2006. We assume that this site may still have some features of plants habitat. And whereas in the absent group, we saw an outlier where we did not see. So this is the place where we didn't see the plant of our interest during the study site. This is a cool analysis grouping these sites automatically, which makes a lot of sense. So then we did PCA analysis, which is also called principal component analysis, using the present and absence data. So here we see that the green shows the presence and red shows the absence. So both are overlapping. So this is because of the outliers. So when we remove the outliers, we see the both groups are pretty much distinctive. And from here onwards, we analyze our data without outliers. And then we do permanova. So this analysis do permutations, which means it plays with the data 999 times, making different possible arrangements of data to find the difference between the different sites. And we found that the sites where the plant is present is different from the site where it is absent in the terms of understory vegetation. So this is where we confirm our previous graphs statistically. And later we did indicator species analysis. This analysis processes the species diversity data and search for the species which are repeatedly seen at particular location or plots. So the sites where plants were absent were not consistent in the terms of species diversity. That why we only see two species consistently at this site. So as you can see the difference, the green color is where we see the plant, and the red color is where we do not see the plant. So indicator species that are seen with the Texas Screwstem are bristle, stalked sedge, prairie moss, rivenwort, American beautyberry, elegant feather moss, white cushion moss, longleaf spikegrass, fabronica moss, and Carolina bayberry. And the where species were not seen, we found oak and green briars, which makes sense that how these two micro habitats within the large habitat are distinguishable from each other. So let's explore the community structure, starting with the species richness. Species richness refer to the number of different species that are present in a specific area or ecosystem or a community. So here we see the green one represents the presence of Bartonia, and red one represents the absence. So with the presence, we see high species richness in the plots where our species is present as compared to the absent sites. So if we talk about overstory species richness, there was no difference in the overstory species. So which means that the most of things are happening understory. So let's go to the diversity indices. So we calculated Shannon index and Simpson index, and we again see the both diversity indices were higher where the species was present and lower where the species was absent. And again, in the overstory, these diversity indices were not statistically different. So here we are talking about the understory vegetation diversity based on the cover percentage categorized by the growth habit of plant species. So here we see forbs. So if we talk about forbs, the present sites have more forbs as compared to the absent. It has more saplings. So saplings are the small germinating plants that could be resulted from the seeds of trees or some small plants that we were not able to identify. So those all are grouped into saplings. And then we see no difference in grasses, but we see a huge difference in sedges. So absent sites has almost no sedges at all, whereas the present sites were uh, occupied by 50% of area was occupied by sedges. And then we see a difference in ferns, where we see higher ferns presence in present plots. And bryophytes, again, we saw higher percentage cover of bryophytes in the sites where Bartonia, where Texas Screwstem was present. So sedges and the bryophytes makes the major difference between the sites where our species of interest is present and absent. Let's talk about canopy openness. So canopy openness uh, explains how much sunlight is passing through the canopy and reaching the forest floor. And we saw a difference between the presence and absent side, where we see the canopy openness where Bartonia is present is around 16%, and where it is absent is around 12%. So in this canopy openness data, we see some difference on the individual plot basis, where we find that the plots that we made on the edge, so at the individual plot level, so as we move out of the core location of Texas Screwstem and reaches the edge location of where we stop seeing the plant, the canopy becomes thicker. And we have very less canopy openness, merely of 5% here. So this was the major difference spotted in the canopy of core locations versus the edge location of species habitat. So we talk about the ground cover where our plant is present. We have more green surface, we have more live vegetation, we have less dead rooted vegetation, because this area is not dry and it has creeks in it, and most of the vegetation is green because water is flowing continuously, maybe seasonally. And then we have high organic debris in the places where we see Bartonia. And then we have very high bare soil at the places where our plant is absent. So we see no stagnant water or running water source during our study period. So talking about soil volumetric water content, soil volumetric water content was around 25% where the species was present, and it was around 7 to 8% where our species was absent. So soil physiochemical analysis, sorry for this brutal table, it's very confusing, but we saw no differences in soil physiochemical properties among all sites. So we run a correlation between the presence of the species of our interest and the biotic and abiotic factors. We found that where our species is present, there is higher species richness, higher species diversity, higher coverage of sedges, higher coverage of live vegetation, higher coverage of bare soil, and high soil volumetric water content. Whereas it was negatively correlated with the presence of saplings and debris. We saw some evidences of threat around the areas where we find our species of interest. So first of all, it's invasive plant known as Chinese tallow tree. So this plant leaches aqueous solutions when the leaves are dry and starts decomposing, uh, which affects the germination of pine trees. So this is from a study uh, carried on the tallow tree and the pine trees, but it's not related to Bartonia, related to our species of interest because uh, we don't have we have not enough information about our species, and nobody has studied the relationship between this invasive plant and our species of interest. And another is Japanese honeysuckle. So this is a fast growing twinning vine that tends to increase the competition for soil resources and spreads on the trees branches, interfering with the sunlight that reaches the forest floor. So this may also be interfering with the sunlight, but no proof has been recorded because we have limited research on our species of interest. And another, this threat is obvious, the invasive animal feral swine. So as you can see in this picture, this is the core location of Texas Screwstem. And these orange flags are the places where we see uh, the plants of Texas Screwstem. So I found a couple of broken plants in this damage sign by hogs, uh, which is a which is a big evidence of threat that how this feral swines are destroying the habitat of the species of our interest. And another, we saw some unethical human invasions on a negative side where we saw uh, trash cans and bottles. And another, at one of our sites, we saw anthropogenic habitat alteration. Although it was a negative site, so so I've heard that this site was diluted by a lady living nearby. She used to transplant numerous species that doesn't belong to that site, which is a perfect example of anthropogenic habitat alteration for aesthetic purposes. And this could be a possible reason for the elimination of Texas Screwstem from this location. And that's why this site is a negative site now. And another, we see hydrological alterations uh, at, at one of our negative site at Hardin. So this site was located at the bank of a dried creek. So we can clearly see the remains of dry water channels in this picture. So this is where that site was located. And this could be the reason why Texas Screwstem is not here, because the creek has changed its routes because of unknown reasons. So let's go to conclusion. So we conclude that Texas Screwstem prefers a very specific habitat occurring in the baygalls of imperiled West Gulf coastal plains, forested seep southern type habitats. And the biotic and abiotic components of the species habitat were unique and different from the edge locations of the baygalls and from the negative sites where the species has not been seen from last 15 to 50 years. So if uh, if we look at this picture, so uh, the biotic and abiotic components here are different from the edge locations here and from the negative side. So this plant as you see in these pictures, this plant occurs in gently sloping to flat sides with perennial springs and seepages or creeks. And this is usually found on elevated clumps of tree bases, roots, which are associated with mosses and liverworts. We observed a distinctive features of Bartonia's habitat based on our research that includes following biotic and abiotic factors. According to our research, we found that where Bartonia is present, we see 70% of area was covered with live vegetation, 50 from which 50% of which were sedges, 15% were bryophytes, and 30% of bare soil that were representing erosion by running creeks and leading to formation of clumps and sedges, which are the uh, habitats or the living grounds for a species of our interest. And we see around 10 to 15% of canopy opening and 20% of soil, 25% of soil volumetric water content. So these all parameters were recorded during the growing season of the species, and extremely higher or lower values of these parameters were associated with the absence of the species. And we revealed a list of key species that was associated with presence and absence of Texas Screwstem. And we found some major threats that are associated with the habitat, which includes invasive species, either plants or animals, unethical human invasion, habitat alteration, and hydrological alterations. So what can we do in the future? The future approaches could be, we could do some more thorough and frequent research, some spatial and temporal studies that monitor the population trends, demographics, and habitat requirements of the species, covering more baygulls within these imperiled habitats. And we are we do not know much about pollination and reproduction information of this plant, so it is must to study the biology of this taxon. So, and very importantly, the soil microbial analysis. This plant has a minimal root system, which is thought to be required some microbial associations that may monitor the distribution of this species. So this is a very important aspect to study. So, in one of the studies from Gillett in 1959, he says that this plant could be living through a mycoheterotrophy nature, which means this requires some microbial associations, either for its germination or any part of its life cycle. And we need to investigate methods to modify these negative sites into the positive sites based on the species requirements that we mentioned in our study, and that we found that how the presence and absence sites are different. So these parameters could be used to modify the negative sites. And habitat protection measures should be improved and monitored in timely manner. And lastly, the eradication of invasive species, check on anthropogenic hydrological alteration, human invasions, and well-managed pine forestry practices should be considered. So I would like to thank my advisor, Dr. Joshna Sharma, and Dr. Robert Cox, and or Dr. Sharma's lab, Dr. Shrilk's lab, especially Azaz Mahmood, and Dr. Matt Johnson from E.L. Reed Herbarium at Texas Tech University, Jason Singhurst from Texas Parks and Wildlife Department, Joe Ligio, Eric Keith from Raven Environment Sciences Services, and Jeff Reed from Fishery and Wildlife Services. So thank you so much. I will take your question. So this is a cottonmouth snake that I saw during my research. Yeah, I'm ready for your questions. Thank you.
Okay. Sorry, can you hear me now?
Yes.
Okay, fantastic. Excellent presentation. Paul, I guess we look at the chat area for the questions come in? Well,
Yeah, it doesn't look like the chat or the Q&A have any questions at this time, Jason.
Okay, well, actually, I have some questions. First, absolutely amazing project and findings and conclusions. Very impressed with your study and your results. Okay, wait, well, I guess we do have a question here. Okay. What was the soil type found with the spruce stem? Was there a specific soil type on the present populations that was consistent, I guess, among the 3 different study sites?
So we do not see much differences in soil physiochemical properties. So, but the soil type was sandy to sandy loam soil, which is, I think, which is common throughout the East Texas because of erosion and the soil is moving. Yeah, sandy soil mostly.
Okay. Let me scroll down here. Give me just a second. What time of the year did you conduct the surveys? Was it during the phenology, like reproduction time? Or?
So it was in October. Yeah, so the plants were there with the flowers. And yes, this was the reproduction time of the plant with the, or we can say the flowering stage of the plant.
And I'll add to that, Sharon. Yeah, this particular plant species is a very narrow phenology, a very like 6-week window.
Yeah,
Like
exactly.
from bolting up to producing its little flower buds to going for and actually producing seeds. So it's you can start seeing populations in September, early October. Really, the month of October through about the first week or 2 of November is the short window this plant comes up and reproduces. Is that about right?
Yes,
Okay.
yes, very short window.
Okay.
Yeah, just 2 or 3 weeks, and then it's gone.
Let's see here. What are the taxonomic or ecological differences between this subspecies and the others? Oh, that's a great question.
So, uh, so basically, these. So this is Bartonia paniculata, and our species of interest is subspecies Texana. So the difference between subspecies paniculata and subspecies Idandra is that this species does not open its flower completely, and it's very tightly packed, and its leaves are very small, very scale-like leaves. Its stem is strictly erect, unlike other subspecies of Bartonia, and it has ovate acuminate corolla lobes,
Yep.
which are 1.9 to 3.2 millimeter long. So this is how it is distinguishable from the other subspecies and recognized as a subspecies of Bartonia paniculata.
Yeah, and I will add that to everybody out there. The subspecies paniculated, the one we see typically the other subspecies that I see in East Texas anyway, is an open herbaceous bogs, much taller plant. It also does kind of a twining and twisting, and actually has the way it does its stem is quite distinct as well. In addition to the very fine characters that you just presented. So
Yeah,
uh,
so I think that's why it's called Screwstem.
yeah.
Yeah.
Okay, regarding canopy openness, did you test during spring versus winter to count for leaf fall? Can you repeat that? Or if you got that?
Yeah, I got it. So no, the work was only done in October, so we did not compare the canopy opening in the season wide. Yeah.
Gotcha. Good question now. Let's see here. Have the seeds been successfully grown in a lab setting?
No. So this was the only field study. We have no permission to pull out the plants. So if you see in this picture, these photos are taken in the field on a white paper and a clipboard. Uh, so I accidentally pull out the plant and then take photos and then put it back to the soil. So yeah.
And that probably had to do with permits with some of the federal
Yeah,
too, yeah.
exactly. And but this time we saw the largest population that has ever recorded. We saw around 200 plants at one of our positive site in Hardin, and 170 plants at Turkey Creek in Tyler. So this was the biggest population ever recorded.
Well, you have me beat, my friend. I've never seen more than 30 plants at the few sites I've seen. Yeah, it's really rare. Let's see. Okay. I think that's the questions I see here in the chat that have come in. It sounds like this is a question from a conservation standpoint. And you did a great summary in your conclusions there. So the question I have is it sounds like in order to, if conservation opportunities of baygulls existed, say with private timber companies in East Texas or individual private landowners that had these baygall habitats, it sounds like you would really want to look at and field check canopy, the subcanopy flora,
Yeah.
did I have this right? The number of sedges and sphagnum and mosses seem to be some of the key characters that if there was potential for this species, you'd want to make sure that those different ecological niches occurred. Is that correct?
Yes, that is correct. So we see the 70% of live vegetation and more sedges and more bryophytes that are associated with this species. Yes,
Okay.
that's correct.
Well, that's wonderful because we have some opportunities in East Texas and ongoing, and I know we'll have future opportunities to hopefully conserve this rare habitat. This definitely helps our program and a stronger rare species botanist and I with your study, and your results are amazing. And to be able to key in on what the key characteristics are that either currently have this species persisting in populations, or what we would need to look for in the field.
Yeah.
Yeah, fantastic. Yeah, so I think that's. Oh wait, I have one more thing here. What do I have here? Oh, yeah, canopy cover. I thought that was quite interesting. The canopy cover was just enough in your canopy cover analysis was just, I think it was like 16% canopy cover or
Yes.
the canopy. Seems like that would be really crucial. I guess my question is, does it seem like that to you in your study that's one of the crucial factors for having just the right sunlight and to have the subshrubs, the ferns, and the herbaceous and the perennial flora. That's part one. Part two is my question, which is, do you think that these habitats go through sort of successional where they close in, and then fire may actually play a role, especially around the fringes, um, to reopen an opportunity for those characteristic plants that are part of its indicator and its association, I guess. What I asked there.
Yeah. So I have not looked into the fire history, but this is very interesting. If we use some fire to open up the canopy and just let the ground surface reestablish, it may open some floor for the species of our interest to grow. And this could be another study area where we can focus on to how to uh, increase the population of this species. And another thing that we talked earlier, the seeds. So uh, if we get the seeds of this plant and just try to germinate them in lab, and then do again transplant them into the field and do a study, carefully monitor, and then study over time, over a couple of years, and this could help to establish new populations of this species.
Awesome. Thank you for your answer to that question. Um, and lastly, the microheterography. Did I pronounce that
Yeah.
right? Okay. So it sounds like the microflora, or sorry, microbes.
Microheterotrophy. Yeah.
Yeah, seems like that's another key component that you, as a conclusion or a resolution of the study, that's important at these occupied sites. Are you still there?
Yes.
Okay.
So yeah, so this is the very important area to search. So there could be, so like in orchids, so orchids need some mycorrhizal fungi to germinate. So if there's no mycorrhizal fungus in the soil, the orchids will not germinate. So it could be similar with our plant. So we need to search for the mycorrhizal fungus or any other microbes that are present in the soils where the plant is present versus the soils where the plant is absent. So mycoheterotrophic plants are form relationship with mycorrhizal fungi, which acts as intermediaries between the plants and the photosynthesis of the organism. So these relationships has a potential uh, to discover, and this may help establish the newer populations of this species if we can find if anything is happening down there in the soil.
Yeah, thank you. That's what I was hoping you would present. Thank you so much, Jarndeep. Well, I want to thank you for your amazing presentation and all the hard work, and also our relationship with our program with your major professor, Jasa Sharma, under your master's study. Absolutely amazing conclusions and results, and we're excited about using your data research to help conserve the species more in Southeast Texas moving forward. And anyway, I wanted just to thank you today for coming here and being part of our webinar series. So we're going to now, I guess, Paul, conclude with the slide. Yeah, here we go.